Okay, well, I'll now commence the launch event for the magnetic particle imaging facility at Monash University. Welcome, everybody. My name's Gary Egan. I'm your MC for today's opening event, and I'm very delighted that you've been able to join. I'd like to commence by acknowledging the people of the Kulin Nations on whose land Monash University is positioned, and I pay my respects to their elders past and present. Magnetic particle imaging is a non-invasive imaging technique that detects superparamagnetic iron oxide particles in vivo. It's a technique that uses changing magnetic fields to generate a signal from the nanoparticles. And since these particles don't occur naturally, it is only the signal of the detected particles that we can image. It's a very excellent new imaging technology. It was first conceived in 2001 by scientists working at the Royal Philips Research Laboratory in Hamburg, and the first system was built in 2005, and commercial preclinical scanners have only become available in the last 4 or 5 years. So we're delighted to be opening this facility, and I'm going to now show a short video of the Alfred Research Alliance Monash Biomedical Imaging facility and the MPI system. This video has been produced by the RMIT facility manager, Dr. Robert Vipul. So I'll now share my screen and show you our video. Let me just try that again. There we go, there it is.
Welcome to the Alfred Research Alliance Monash Biomedical Imaging Facility, which is located here at the Alfred Hospital Precinct. The newest addition to our preclinical imaging facility is the magnetic particle imaging system from Magnetic Insight, which is equipped with a CT and a hyper platform. The combination of these 3 modalities makes this setup a world first, allowing for complete characterization in one single facility.
MPI features high imaging contrast, high sensitivity, and 0 signal depth attenuation. That makes it ideal for my cancer research to detect even the smallest metastases in vivo. I'm fortunate to work on the world's first MPI system featuring an inbuilt computed tomography system. The Hyper system is a modality which can apply therapeutic heat on a localized area. The temperature can be moderated and set to a cancer-damaging rate. The synergy of both modalities, MPI and Hyper, will play an important role in the new field of Theranostics.
The addition of magnetic particle imaging was made possible with funding from the Australian Research Council, Monash University and RMIT University.
So unfortunately, as a virtual event, that's all you get to see of the facility, but thanks very much to Rob for making that great video. So I'd now like to proceed on with inviting our first guest speaker today, Professor Mike Ryan, to officially open the facility. Professor Ryan is the Academic Director of Research Infrastructure at Monash and Head of the Mitochondrial Biology and Disease Laboratory in the Monash Biomedicine Discovery Institute. He joined Monash in 2014 and served as the Deputy Dean, Research and Research Infrastructure in the Faculty of Medicine, Nursing and Health Sciences until earlier this year. So I'm going to hand over to Mike. Thanks, Mike.
Thanks so much, Gary, and thank you everyone for joining us. And I'm really delighted to officially open the magnetic particle imaging capability at Monash University. As we've seen, the MPI is a new non-invasive imaging technique with significantly increased sensitivity over MRI and faster acquisition times than PET and MRI. The capability at the Alfred Research Alliance and the Monash Biomedical Imaging site is the world's first MPI system with computed tomography and hyperthermia capabilities, providing unique capabilities that open the door to these cutting-edge opportunities for interdisciplinary projects in medical research and chemistry and biotechnology. The MPI system was developed by Magnetic Insight, funded by the Australian government through a LEAF grant from the Australian Research Council, which we greatly appreciate, and from contributions both from Monash University and our colleagues at RMIT University as well. The MPI system is accessible to all researchers and industry across Melbourne and Australia, and we welcome, obviously, researchers to utilise that facility. It will further enhance Monash's leadership and expertise in advanced material science, nanotechnology, molecular imaging and biosciences. And I really look forward to hearing how this instrument will actually support the cutting-edge research activities and driving impact from our speakers that we'll hear about today. So thank you very much, everyone, and thanks, Gary.
Great. Thank you, Mike. Great to have the facility officially opened. It has been a bit of a journey getting the facility established. We had the equipment first delivered in April last year, and of course that was at the beginning of lockdown, so it was extremely challenging to have it installed and make it operational. But I'd now like to hand over to Professor Callum Drummond to speak on behalf of RMIT University. Professor Drummond is the Deputy Vice Chancellor, Research and Innovation and Vice President of RMIT. He joined RMIT in 2014 from CSIRO, where he was the Group Executive for Manufacturing Materials and Minerals. Thank you very much, Callum.
Thanks, Gary. And I'd like to begin by acknowledging the traditional custodians of the land on which we are gathering virtually today, especially to the people of the Woiwurrung and the Boonwurrung language groups of the Eastern Kulin Nation, from where I am joining the event today. And I pay my respects to elders past, present and emerging. It's my great pleasure to attend this official virtual launch of the Magnetic Particle Imaging Facility this afternoon. RMIT University and Monash University have a strong history of collaboration, and we continue to engage to jointly build critical research infrastructure for Victorian and Australian communities. Over the past 5 years, through the support of the Australian Research Council's Linkage Infrastructure Equipment and Facilities Scheme, otherwise known as LEAF, RMIT and Monash University have successfully established 18 joint infrastructure capabilities in areas of high-speed electronics and optics, functional materials, aerodynamics, robotics, and materials and biomedical imaging. These facilities are then efficiently utilised by joint projects and researchers across our two universities in particular, leading to improved outcomes and specifically improved outcomes for our early career researchers and our higher degree by research students. The MPI facility is one of the latest outcomes of this collaboration. The facility will be critical for the generation of new knowledge in the industrial biosciences research sector and provide unique benefits to the national research community. RMIT has a strong track record of research in IP in the area of magnetic nanomaterials and contrast agents, having successfully licensed one of our biomedical imaging products for tracking of immunotherapeutic cells in cancer patients to an Australian biotechnology company, Cell Therapies Proprietary Limited. So RMIT researchers are well placed to drive innovations in the development of new contrast agents with the MPI. I would like to congratulate the MPI team, in particular you, Gary, and our own RMIT Professor Vipul Bansal for taking the initiative to establish the MPI facility in Australia. I wish them and the team all the best for your future research endeavours. Thank you, Gary.
Good, thank you very much, Callum. Look, it is great to have RMIT University as a scientific collaborator and a financial contributor, a major financial contributor to the facility. We really have appreciated that, and you know, there's been terrific past collaboration, but I think this is really going to take it to the next level. So we really look forward to strengthening our cooperation with RMIT. As we've heard, the facility has been funded by the Australian Government through the Australian Research Council, a LEAF grant that was awarded in 2019. Extremely grateful to the ARC for their support in that regard. And I would now like to introduce Dr. Robert Munn, Executive Director of Engineering and Information Sciences at the ARC. Prior to joining the ARC, Dr. Munn was branch head of the Defence Science and Technology Group within the Australian Department of Defence. His research expertise is in the area of fluid mechanics and complex systems integration, with a focus on integration and networking of multiple defence systems. So we hope, Robert, that we'll be able to turn our attention towards some of those goals that we have nationally. And thank you very much for joining us today to help launch this facility.
Thank you, Professor Egan, and good afternoon to everyone. I would also like to respectfully acknowledge the traditional owners of the land on which we're all meeting today and pay my respects to elders past, present and emerging, and I'll extend that respect to Aboriginal and Torres Strait Islander people present today. I'd also like to acknowledge all the distinguished guests, researchers, students and partners here or online at this event today. I'm here representing our Chief Executive Officer, Professor Sue Thomas. Professor Thomas sends her apologies and her best wishes for today's event. The purchase of this impressive facility has been possible through an award won by Professor Egan and his team from the Australian Research Council, funded by the Australian Research Council with additional investment from Monash University, RMIT. The RC Linkage Equipment and Infrastructure and Facilities Program, or LEAF as it's commonly referred to, seeks to provide cooperative initiatives for the acquisition of mutually beneficial research equipment. It encourages institutions to develop and collaborate to develop a collaborative arrangement such as those we've seen here today between Monash and RMIT. And I note that this is one of our oldest schemes and has been supporting the acquisition of hundreds of items of research and infrastructure at universities for over 20 years. I'm pleased that LEAF has now supported the acquisition of the first magnetic particle imaging facility in Australia. This is a breakthrough technology with applications across fields and diverse disciplines, such as cell tracking, materials science, and biotechnology. I particularly congratulate Professor Gary Egan and all the research teams involved with this successful collaboration. I understand there will be synergies with the work of the two ARC Centres of Excellence based at Monash, which are led by CIs on this LEAF grant. They are the Centre of Excellence for Integrative Brain Function, led by Professor Egan, and the Centre of Excellence for Convergent Bio-Nanoscience and Technology, led by Professor Tom Davis. This new MPI facility will enhance the work of these two ARC centres and help establish Australia's leadership in nanotechnology and molecular imaging. The ARC and I look forward to hearing about the important insights and outcomes you produce and look forward to some articles in some of our future publications from you. So thank you all for inviting me here this afternoon to see what you're doing.
Great, thank you very much, Robert. We really appreciate the ARC's support for research nationally and particularly this facility. The university has spent the last couple of years establishing a strong relationship with the supplier of our MPI scanner, Magnetic Insights, based in the United States, as well as their Australian distributor, Trend Bio Proprietary Limited. And it's a pleasure now to welcome Trent Warburton, who's the Managing Director and founder of Trend Bio, which is a scientific instrument distributor and manufacturer based in Melbourne. Trent's been involved in life sciences research sector for more than 20 years across disciplines including imaging, genomics, proteomics and cell biology. And he's got key interest in the commercialization of technologies for use as research tools. So we're really delighted, Trent, that we've been able to partner with yourself and Magnetic Insight. And I'd like to invite you now to say a few words at this opening.
Yeah, thanks very much for that, Gary. First of all, I just wanted to thank a few people involved in delivering this project. So I'd like to thank Robert Bajalka, who's been instrumental from Monash's side. Robert, as the facilities manager, has really helped the Magnetic Insights team and our team in delivering this project. Also, Ashley Truxell and Julie McCoy from Ashley from Magnetic Insight and Julie from our team, both physicists who have been involved in delivering this. So there's been a lot of early mornings for Julia and late evenings for Ashley. So I thank them both for being able to deliver this in a year where they couldn't travel. So delivering a world first facility here in Australia has been a big effort from those guys. So thank you very much. Our experience as a distributor and manufacturer has always been that we've seen some great research collaborations in Europe and the US. And so we've really pivoted locally to try and engage with the research community the same way. So early on in this process, we were really enthusiastic to work with Gary's team the same way. And we were really happy that they embraced a collaborative approach with Magnetic Insight and ourselves. So these sort of collaborative agreements, whilst we've heard of the agreements between Monash and RMIT, an agreement with a commercial partner offers some unique advantages to the university and also the commercial partners. What it can deliver in terms of research outcomes is it can give you access to developing technology. So when, as a developing imaging modality, MPI, there'll be new technologies developed. So hopefully Monash will be able to access those, whether it be consumables, hardware, software. It'll also give them access to the R&D scientists from the factory as well, to hopefully deliver really great research outcomes for everybody involved. From the commercial side, it also gives us access to Monash's feedback when it comes to new product development, generating new applications and working with the new software updates and different consumables that'll hopefully drive new markets and real commercial outcomes for us as well. The other thing these kind of agreements and collaborations do is that it opens up different funding and investment opportunities that exist when you have a commercial partner on board. So we're excited to see where that will take us. And as a local company here in Australia, it also gives us a good window in what's happening at the lab level at Monash as well. So different innovations that are happening there that may be completely not related to any kind of collaborative agreement that we have, but there may be other commercialization opportunities that, that exist once we get more of a window into what's happening at the research lab level. So I want to thank Gary and also Patrick from Magnetic Insight for enabling this collaborative approach, which will hopefully see some really great research outcomes in the future, and there might even be some commercial ones as well. So thank you very much.
Great, thank you so much, Trent. And thanks to both Trend Bio and Magnetic Insight for their support for this launch, which you've both contributed to. We appreciate that. Also your commitment to the establishment of the Monash MPI facility as a centre of excellence for magnetic particle imaging. We're going to be really, I think, able to do terrific things in that regard. And I'm also really pleased, Trent, to hear your comments about a broader cooperation agreement between Monash and Trend Bio. I think that'll be really exciting to explore further once the MPI agreement is operating. So finally, I would like also to thank the Monash Research Technology Platform staff who've worked so hard to bring this project to fruition. And in particular, I'd like to thank Robert Belgica again for his great video, but also the incredibly hard work he's been doing over the past 15, 18 months in establishing, preparing the Arambi facility, establishing the MPI facility in place, and then, as Trent explained, working all different hours with the factory people over in Alameda in California to make this operational. It's been a real tour de force. I'd also like to thank Kylie Reid, who's the General Manager of MBI Research Platform, for her support for this project and her team's support for this project as well over that period of time. It has been a real team effort to bring this together. So thank you, and thank you to all of our presenters here and comments made this afternoon. We really appreciate that. That now concludes our opening speeches. So thanks for your attendance, everybody. We're now going to move on to the scientific symposium. We have 4 terrific presentations, which we're going to now move into. So we're now going to the scientific session. Glad to see everybody staying online. I understand if you're not able to, but please do, if you can, stay on board with us. So our symposium this afternoon is going to involve 4 speakers, and our speakers are Patrick Goodwill, who's the founder and CEO of Magnetic Insight. He's going to be then followed by Associate Professor Christoph Hagemeyer from the Australian Centre for Blood Diseases at Monash University. We'll then have a presentation from Professor Vipul Bansal, who's Director of the Serene Potter Nanosensing Facility at RMIT University, and a presentation from Dr. Karen Alt, who's also at the Australian Centre for Blood Diseases at Monash University. So I'm really looking forward to these presentations. I think we're going to get to see some of the first data coming off the MPI scanner as well, which will be terrific. We're going to invite questions from our audience, and those questions need to be entered into the Q&A panel. And then at the end of each presentation, we'll have a question or 2, depending on time. And then at the end of the 4 presentations, we'll go back into a further discussion between the panelists, as well as taking any further questions that have come in. So we look forward to your questions coming in. Please make them nice and succinct, and then we'll get our presenters to address them. So our first presenter is Dr. Patrick Goodwill, who's a world expert in magnetic particle imaging and co-founder of Magnetic Insight. He has his PhD from the University of California in Berkeley and UCSF, the joint graduate group in bioengineering, with a bachelor degree and master's degree from electrical engineering at Stanford. He's co-authored over 70 papers and has over 20 patents in this area. Prior to founding Magnetic Insight in 2015, he designed and built 5 prototype MPI scanners and developed the theory of X-space image reconstruction for MPI. So we're really delighted, Patrick, that you're able to join us today from Alameda, from the factory, and we really look forward to your presentation. Thanks, Patrick.
Well, thank you so much for having me here. Let me share my screen here. Let's see if that works. So thank you so much again, Professor Egan, for having me here today to be part of such a wonderful event. And as the earlier speakers mentioned, the Monash site is the first commercial MPI and CT system, and the first to also include that hyperthermia system as well. And I can say without reservation that this is the most advanced system we've ever installed. And I also wanted to thank you for your trust in us and express our excitement to work with you and your team to build on the cutting edge of both biomedical imaging and the MPI field. I really can't wait to see where we go with this incredible technology. What I'm going to speak about today is magnetic particle imaging fundamentals. So my goal here is to get people on board with where, what MPI is, how it works, and then some of the other speakers are going to speak later on the applications of it. So I'm going to start with the MPI physics, really how it works, what the tracers look like on a very general sense. Then I'm going to go into the hardware, and finally a very, very brief application, tour of applications, and really kind of an overview of what some of the other people in the field are also working on. So as we jump into MPI, the way we think of it is it's a safe and accessible cellular and tracer imaging. What I mean by that is we see a tracer, and that tracer is an iron oxide typically, and we see it with incredible sensitivity and specificity. And this gives us a lot of applications across diagnostic and therapeutic areas. And as the systems start to improve, we have preclinical systems commercially available and clinical systems currently in development. On the right here is sort of a representative MPI CT image. The MPI image is in blue and gold, and the CT is in the background for co-registration. So how does MPI work? To understand MPI on a very fundamental level, there's 3 concepts to understand. The first concept is, how do nanoparticles respond to an applied field? The second thing is, how do we localize nanoparticles with what we call a selection field? And finally, how do we detect those nanoparticles? So if I have nanoparticles, like you saw on that previous page, those little round nanoparticles, they each act like little bar magnets. And when I put them in a room where there's no magnetic field, they randomly orient because thermal energy knocks them around a little bit. If I say add 2 little current loops over here and turn them on, and I produce a field with these field lines, the little bar magnets essentially align with the applied field. Now, if I reverse the current in those little loops, the bar magnets are going to align in the opposite direction. So this is how magnetic nanoparticles respond to a magnetic field. They align with that field. Okay, so the second concept is, how do we make a specially shaped field, which we call the selection field? So if we have these 2 magnets here, these are actually permanent magnets. Say the north side is on this side, north side is on this side. If we draw the field lines look like this, where they wrap all the way around actually. They don't just stop right there. They wrap around at the back of the magnet. And at the very middle of these 2 magnets, we have what we call the field-free region. Now, the field-free region is where the magnetic field passes through 0. Essentially, say this side over here is at, say, -0.5 Tesla, and this region over here is +0.5 Tesla. Right in the middle, it's at 0. So if I were to take some magnetic nanoparticles and just place them in these different regions, if I place them in A over here, the magnetic particles are going to align with the field lines, point to the left, and so they're all aligned like this and saturated in the negative direction. place particles in the middle in the field-free region, and they'll randomly orient again from that thermal energy. Finally, on the far left, particles are going to be pointing to the right. So this concept right here really underlies a lot of what's happening in MPI. And we've done something that's very amazing. We've managed to control the direction of nanoparticles from a distance. So even though these magnetic nanoparticles are here in the middle of this region, and this field-free region might only be a millimeter across or smaller, we've been able to control the direction of those nanoparticles using these magnets that are, can be centimeters to, at 10, 15 centimeters away in a larger system. Okay, so that's the second concept. Now for the final concept is, how do we produce a signal? So if I have a nanoparticle here and the field-free region here, if you notice, the nanoparticle aligns with the applied field, and then I'm going to shift this field-free region over the nanoparticle, and the nanoparticle is going to flip. So essentially the particle is pointing to the right over here, now it's pointing to the left. And when it goes through that transition, we detect a signal from the change in magnetization, amplified by a preamplifier. So it's as simple as that. Now, how does it work in 2D and 3D? So if we add a few different shift coils, if this is the magnets that produce the field-free region, and then we shift it around with these homogeneous fields, and then we detect a signal. So if we shift like this, where the field-free region is doing a raster pattern, the signal that comes out is actually this strange looking signal over here. But if we take this signal that we receive and we grid it to the instantaneous position of the field-free region, we get an image of the nanoparticle. And the images that come out really do look like this. We call it almost like a bow tie. So there's a nanoparticle in the middle, you get kind of this bow tie signal that comes out. If we shift rapidly in this other direction, the bow tie changes direction. So let's put this all together. So these are the permanent magnets, or electromagnets rather, and these electromagnets produce a field-free region. And the field-free region I'm going to show as a yellow line here, and this is actually a field-free line in this case. And whenever the field-free line is over the magnetic nanoparticles, when it passes over those nanoparticles, they're going to flip to follow the applied field. We're going to detect all of those signals that come out, put it together with the math, and we're able to get an image of the nanoparticle concentration across the animal. And this is an actual MPI image of the blood volume in a rat head. So MPI is very unique among imaging modalities because the tracers change the sensitivity and the resolution of the system dramatically. And we're able to measure this using what we call a relax module, which is a relaxometry module that gives essentially the point spread function of a nanoparticle in a non-imaging format. Then of course you can actually image the nanoparticles in the imaging format and see if they match. There's a lot of opportunity for optimization in nanoparticles. Here's some new nanoparticles being developed by the University of Florida and UC Berkeley. And these particular nanoparticles are acting in a very strange way. They're acting ferromagnetically. And so they're able to have up to about 200 micron resolution with no change in hardware. It's going to be a while till we're able to get these into solution even. So there's a ways to go before they make it into animals. Question I commonly get is, what's the difference between MRI and MPI? Well, essentially, MPI scans cannot be performed in an MRI scanner because they're fundamentally no relationship between the two technologies. An MRI has a homogeneous field, whereas MPI has that gradient field. And furthermore, the MPI images that come out are what we like to call positive contrast, where the iron oxide shows up as a bright signal, which is contrasts with, say, the exact same animal in an MRI where the iron comes out as black. Something that we'll talk about a little bit later is MPI can do more than just imaging. So what we saw in imaging is if the field-free region is here, the nanoparticles can freely rotate at the imaging frequency. But if we, say, put a much higher frequency, say at 340 kilohertz, the nanoparticles will heat up. As we, furthermore, with this field-free region, if particles aren't at the field-free region and we apply the RF field, the particles will just wiggle a little bit and they won't heat. So this lets us selectively choose where we heat. And this is part of what the hyper system that we're talking about, is that we're able to prescribe a heating regions, and then we're able to selectively heat them using the selection field gradient. Let's take a look into what the hardware looks like on the inside of the MPI system. First off, as an overview, the momentum CT, the momentum imager is in the back, and then the CT is in the front. And here's a representative image from our CT. And here's the team that made it happen. So when we're building MPI systems, we have a field-free region, and the field-free region can be either a point, which is a point in 3 space where we can get a signal, or if we stretch it out, we can get what's known as a field-free line. Interesting thing about a field-free line is that it has 10 to 100 times more volume, and because of that extra volume, we're able to get a lot more sensitivity. And so to maximize sensitivity and speed, we actually use a field-free line in the system that's in the momentum system. To produce an image, the first thing that we do is we create a field-free line, and we use those permanent magnets as an illustration here. In the system itself, we have an electromagnet. And then we slowly translate the field-free line across the entire sample. It turns out that the RF field, if we were to try to make it large enough to translate the field-free line across the entire sample, would cause too much heating. And so we actually do it slowly. Then, on top of that slow transition, we add a fast transition, and that's what gets the large MPI signal out of the nanoparticles. We receive that signal using a receiver coil, and then to get a tomographic image, we rotate and repeat, and we use standard projection reconstruction to produce a 3D image. So it's taking a closer look at the hardware itself. Here's the imager, and as we get closer, here's a plastic mouse. And the system has a 2-stage slide. You push the slide in manually, and then the motor takes over after that. Taking a closer look inside the instrument, as we open the side here, you can take a look at the main magnets. These are copper electromagnets. It's got the blue and red. That's cold water coming in and hot water coming out. It takes about 30 kilowatts on average. And then, of course, as I described before, the whole main magnet rotates. On the side here, that's iron, and this is all a stainless steel gantry to be able to not interact with the electromagnets. Inside the system are cooled RF coils. This is an older version. These are water cooled. The latest version that Monash has is actually oil cooled because of the high voltages that are used in the system. And importantly, we have no wires on the floor so that people can mop around the system. Because people can't really see the system at Monash right now, I've put in an example system for your reference. The Monash system looks a little bit different because there's a CT on the front, whereas this one doesn't have a CT. So getting a little bit into the applications, we have customers that are working on cell therapies, vascular perfusion, inflammation, theranostics, nanotechnology. So it's a very wide range of different applications. They range from University of Florida, who's tracking T-cell immunotherapies, Robarts tracking circulating tumor cell homing, all the way over to Michigan State, where they're looking at nano carriers that when they break down, the MPI signal turns on. There's actually a lot of very unique things that are being developed. And if we try to classify all the different projects that we did a survey about late last year, about a quarter of the groups are doing oncology work along with cell tracking. A lot of people are working on nanoparticles and nanoparticle development. A small set of people are working on the Theranostics, and that's the hyperthermia system. And finally, inflammation is another growing area of research. So on the cell therapy, people are doing tracking of either stem cells or T cells, adoptive cell therapies, because what we can do is we can track the location over time. And so, for example, here's a very early experiment from University of Florida where they're tracking T cells tracking and homing to a glioblastoma. And so they're able to see it, make it to the glioblastoma in about 24 hours, and then they start to wash away after that. And so they're able to see the biodistribution and the retention of the nanoparticles and the cells that they're in. And the goal is to be able to see how many cells make it to a site of inflammation or a site of some sort of disease, and then to try to improve our ability to get those cells there over time with different sort of drugs that we apply while administering the cells. Here are some lists of the different types of cells people have tried to detect over the years. And some groups have been able to see as few as 250 cells, which is an amazing feat. So 250 cells in a single voxel. On inflammation, we've been working closely with Stanford to look at tumor-associated inflammation, as well as neuroinflammation, either following some sort of event, such as a trauma event. So this is the Berkeley group doing traumatic brain injury measurements. And the nice thing about measuring inflammation with MPI is you can do it repeatedly. And this is something that we see as a potential diagnostic in the future. So with that, I want to end this presentation with a brief overview of magnetic insight and what it took for us to get to where we are. So over the past few years, we've been very productive. We developed the momentum system essentially in 2017, but we didn't really have a commercial launch until 2019 after we worked out some of the kinks. In 2020, we launched the CT in addition to the momentum. And right now we're developing a clinical scale system that we should be testing in large animals in 2023. On the sales side, here are a number of the places that we've installed systems at. I don't break down the momentum and the momentum CT, but Monash, for example, has the first momentum CT and one of the few hypers in the world. And finally, our funding has been very important for doing this. I don't list the NIH funding here, but we've had, I think, 6 or 7 grants from the NIH over the years. And then, of course, venture funding from seed funding, a Series A, and right now we're raising a Series B. So if you know anybody who wants to work with us on a Series B to reach first in man, please send them over. So a brief overview of our different products. So on the preclinical side, we have the Momentum CT and the Hyperthermia system. And the goal for these systems really is to establish the imaging market for MPI. And our customers are customers like Monash, where they're researchers and they're working with some early systems and prototypes like the Hyperthermia system. And right now, this is where we're transitioning and focusing is on the clinical imaging for a very large MPI market where our customers can be radiologists. And it has quite a bit of development, but the future looks very bright for MPI. So a quick look at some of our customers around the world. And again, thank you very much for having me. And I need to acknowledge some of my collaborators over the years, including Steve Connolly over at UC Berkeley, Carlos at University of Florida, Chris Contag at Michigan State, Paula Foster at Robarts, and our Stanford friends, and of course, our members of the company. Thank you very much.
Lovely. Thank you very much, Patrick. Maybe, yeah, if you can stop sharing, that's great. So I encourage people to put any questions they have up in the Q&A. I'd like to just ask and start the questions, Patrick, really to ask you where you think there is, I guess, the real need at the moment to further advance the MPI. We were aware when we started looking about the need for co-registration, and you've now addressed that with the CT. I think contrast agents and nanoparticles is clearly a very active area. What's your sense of the most important areas that need further development in a research context?
Yeah, that's a really great question. I would put it on 2 major focuses. The first is on the application itself. So there's a large number of applications for MPI, especially in adoptive cell therapies like T-cell therapies and immune imaging. And that takes a lot of work to get to the point where we're very confident in it. And so I see a lot of work that can be done there. And the second one, of course, is on the tracers that make the applications work. So for example, we're starting to see people that are making nanoparticles that have antibodies on the outside that are able to label different types of immune cells in the body. And so we're able to see rare cell populations throughout the body and get really a map of them. And so we're just starting to see the barest of trials on those. And I would really love to see where over the next few years we can bring it.
And it's very exciting to see your roadmap with your clinical MPI system slated for 2023. It's not far off, actually. That's very exciting. Can you say a little bit more about that?
Sure. So that's the result of I think we're on year 5 of a NIH-funded project to get the system to a clinical scale. So we'll be doing essentially a neuroscanner. And so we're targeting, well, essentially we like to think of it as we like to walk before we run. So we're doing something that's a neuro-sized bore, I think is 23 to 25 centimeters. And that's intended for both large animal research and neuro testing of both perfusion and adaptive cell therapy tracking.
Very exciting to know about that. So thank you again, Patrick, for that fascinating and very informative presentation. And we look forward to working with Magnetic Insights over the coming years.
Thank you for having me.
Okay, so we'll move on to our next presenter, Associate Professor Christoph Hagemeyer. Christoph is an Associate Professor and a Senior NHMRC Research Fellow and Head of the Nanobiotechnology Laboratory in the Australian Centre for Blood Diseases at Monash University. His expertise is in the application of small recombinant single-chain antibodies for molecular imaging and drug delivery. And his research focuses on the development of antithrombotic fusion proteins and novel imaging probes for the treatment of cardiovascular disease. Christoph was really the power and the driver behind the grant application, so I do want to acknowledge Christoph's absolutely leading role in the establishment of this facility, and also the exciting research that he's going to share with us about using iron oxide particles in MRI and how we can learn from that to develop optimal iron oxide particles in MPI. Thank you very much, Christoph.
Thanks, Gary, for the nice introduction. I share my screen. I hope you can see that. I want to get my laser pointer. So as Gary said, what I want to talk about is really what we have done over the last 10 years or so on targeted iron oxide particles for the diagnosis of cardiovascular disease. And I hope you appreciate that while I can't show any MPI results with the technology, you hopefully can appreciate how these technologies we have developed can be used in MPI in the future. So why we're interested in cardiovascular imaging and diagnosis, it's really all around atherosclerotic plaques, vulnerable plaques. They can't be identified currently, so there's a continued mortality and morbidity from heart attacks and strokes. And they come at a young age and often without any warning. So there's an example here of a businessman which has mild risk factors and suddenly he develops chest pain and has a heart attack. So that's really the disease we want to tackle. And this problem is not going away anytime soon. It's rising everywhere, more so in low and mid-income countries. So if you look here at the red bars, so the developed countries, of course, have overall a lower mortality. But if you come to the mid and low-income countries, there's a dramatic increase in cardiovascular disease burden to is expected in the next 15 to 20 years. So what's the, sorry, go back. So it's now well appreciated what the underlying morphology or the disease morphology is. It's really the atherosclerotic plaque and more particularly the outward modeling soft plaques, which don't show up in classic angiography. So when you do a CT with a contrast, you usually see a well-perfused vessel, but there could be unstable plaque hiding in the vessel wall, which can't be detected with conventional imaging. And that really is a remaining challenge to identify these vulnerable plaques, because they are really causing the majority of myocardial infarcts. So platelets is a main focus of my lab, and platelets are central to cardiovascular disease, and they are a hallmark of vulnerable plaques. They're not only a part of the thrombus, but they're also now widely appreciated as inflammatory players and really part of the immune system. So in many ways, they can fulfill tasks predominantly attributed to classic immune cells. They can hunt down pathogens, as shown in this excellent paper a couple of years ago from the Marsberg group. As you can see that platelets can sense pathogens and track them down and also collect these pathogens here in this video. Platelets are also outnumber most of the other immune cells. So if there's something going wrong in the body, it's usually platelets who arrive first. and they stick to really everything except for healthy endothelium. So anyone who has worked with platelets would appreciate this statement. We have a long-standing interest in one of the main receptors on platelets, the GP2B3A integrin. It's specific for platelets, highly abundant, so ideal for molecular imaging. And most importantly, it undergoes a dramatic conformational change upon platelet activation, which can be specifically targeted with conformation-specific antibodies. One major way to make these antibodies is, of course, a phage display, which finally was awarded the Nobel Prize to Smith and Winter in 2018. And a bacteriophage is a virus for bacteria, as you probably know, and it combines really the antibody structure on the surface of the phage with the genetic information on the plasmid, which is inside of the phage. And this really allows, through panning, so depletion and selection, to obtain really high affinity antibodies. Unfortunately, most antibodies and also clinically approved antibodies against the target to B3A are binding independent of the conformation. So they will block all circulating platelets and thereby causing off-target effect and bleeding complications. Using a very clever panning strategy, the antibody we have been using in the past only binds to activated platelets. This was achieved by selecting not only between activated and non-activated platelets, but also between CHO cells which had the activated receptor expressed and the non-activated receptor. So by changing the background between CHO cells and platelet, it was possible to select an antibody which only binds to the active conformation of GP2B3A. And here is shown how exclusively this antibody binds to activated platelets compared to non-activated platelets. Initial work with this antibody was done in MRI, but we have also explored this antibody now extensively in PET and also in ultrasound imaging. As you are well aware, all imaging modalities are usually a trade-off between resolution, so MRI has a very high resolution, and CT as well, and sensitivity, where PET is really a strong player. And that's why we're so excited about MPI, because MPI really combines the best of all worlds. It has high sensitivity, good resolution, and high speed. The initial work we did with iron oxide particles in MRI was very simple. You take an off-the-shelf iron oxide particle, you mix it with a coordination chemistry which contains a cobalt ligand, and this cobalt ligand binds to the his tag of this recombinant single chain antibody. So you mix the two and ready is your contrast agent, which is very simple. So it can pick up thrombosis in a test tube. So here you see thrombi, which are in a petri dish, and then you mix it with the contrast agent and you put it in MRI. You see a signal on the surface of these thrombi. But more excitingly, it was also possible to pick up an MRI signal on symptomatic carotid artery plaques. So these are plaques which are surgically taken out of patients who have transient ischemic attacks. And you can see here in histology that you can see these particles binding to activated platelets on the surface of this vulnerable plaque. And also in MRI, you can see that there's an increased contrast on the surface of this symptomatic plaques compared to the control. So this was all very, very exciting. However, the low sensitivity, and Patrick already mentioned that, it's really a problem that MRI shows usually a black hole in a gray background. So you really want to see something which is not exclusively there, like in MPI, where you literally have no background. And that's why I'm so excited to really use this technology in MPI in the future. This technology can also be used to measure thrombosis and thrombolysis in vivo. So this is an MRI in a mouse carotid artery injury, and you can see that with a white blood pool, you see perfusion in this vessel before you inject the iron oxide contrast. And then over time, the iron oxide will bind to this developing thrombus and the signal goes black. And then you inject your clotbuster and you have immediately the reperfusion, and you can monitor that in vivo, which is quite exciting. So overall, very promising proof of concept. However, these particles are very large. They are 1 to 2 micron. The bioconjugation chemistry is not translatable into the clinic with the cobalt conjugation chemistry. And the coating of the particles is a polystyrene. So it's basically a plastic coating on the surface of these particles. So really, we tried over the following years, try to develop a few of these aspects towards clinical translation. The first thing we worked on was the bioconjugation. And that's very important if you want to have a covalent site-specific bioconjugation. Because a lot of people, when they show coated particles with antibodies, they show this picture here, where you have nicely orientated antibodies on the surface of these particles. But in reality, if you have a non-site-specific bioconjugation technique, that's not the case. So the antibodies are typically randomly orientated on the surface of these particles. And that's a problem because only a fraction of these binding ligands will be active. And that's shown here, especially with small recombinant antibody fragments. You get a loss of binding when you not specifically attach something to these antibodies. So we did major work on site-specific bioconjugation with an enzyme called sortase, which is a transpeptidase. And it's a very simple process. You have an LPTG peptide tag on the surface and the C-terminus of the antibody, and then you have a GGG peptide on the surface of the nanoparticle. And you just mix that with the enzyme, and you have a new peptide bond formed by this enzymatic reaction. It's very mild and very efficient bioconjugation. And it works really, really well in vitro and in vivo. So in vitro here, we use flow chamber assays to show the specificity of our targeted nanoparticles. And here on the top, you see the control, and at the bottom, you see the targeted particles. And over time, over 3 minutes, you see with exquisite specificity and affinity, these particles bind to this little trombi, which are immobilized on the surface of this flow chamber. And if you go in and look in confocal microscopy at these plated aggregates with the particles, you see that they stick exclusively to the platelets and the developing thrombus, and it made it on the cover of Circulation Research at the time. That also works very well in vivo. So this is a model. Show that this is a model of intravital microscopy. So intravital microscopy is that you go with the microscope directly onto the vessel and image what's happening in the vessel in real time. And we make an injury at the side of the vessel wall. On the left is the control, on the right is the targeted particles. And you can see that within a few seconds after injecting these particles start to find the developing thrombus and bind to the surface of this thrombosis. So very, very nice results to show the exquisite binding of these particles. And you can appreciate that we are very excited to look at something like this in an MPI system in real time in a live animal. So that should be really, really interesting to develop that further. I can't disclose what we're doing in this space because it's a non-confidential forum, of course, but we are working on novel particles. We are working on novel surface coating of the particles, and then also on the bioconjugation aspects to really tailor-make particles for future use in MPI. So in summary, we have demonstrated that platelet targeted iron oxide particles are versatile MRI contrast agents for the detection of thrombosis, inflammation, and the monitoring of thrombolysis. Poor sensitivity and concerns about the high dose of contrast required for MRI and the low signal to noise limits the clinical application of this in MRI. With the higher sensitivity requiring less contrast and no background, MPI is really a breakthrough technology with enormous diagnostic potential for vascular diseases. Our facility here at Monash is the most advanced MPI center in the world now, combining the momentum CT and the Hyper system. That leads me to thank my group, my international and local collaborators, funding bodies, especially the ARC for the generous award of the LEAF grant, which made this facility possible. And you can follow us on social media if you like. Thank you very much for your attention.
Great, thank you very much, Christoph. And Christoph, I have been advised that you're actually a professor, so congratulations on that appointment as well.
I'm not sure if that's official now. I think it's effective early July, but thank you very much.
Thank you on an email.
Yeah, this was a nice surprise yesterday.
Oh, terrific. Very richly deserved. It was great work you've been doing. I was looking at those intravitreal microscopy images that you showed, and they're obviously at very high resolution, as you'd expect with microscopy. What do you think the resolution is that you'll need to really nail these sort of problems in mouse models?
Yeah, that's a very good question. I think we know that it works in MRI, as I showed with the thrombosis and thrombolysis. So that was basically MRI looking at white blood pool, and then these particles on the surface of the developing thrombus shining into the entire lumen and quenching the signal. But these are 1 to 2 micron iron oxide particles. They have a huge field which influences a wide area in this field. With MPI, we have to see. I think the sensitivity will potentially be higher, but yeah, we don't know. But we get as you can appreciate from the intravital, we get a fair bit of particles accumulating there in a very small area. So I think the target abundancy is quite high.
Mm. And the preparation of these nanoparticles, you were talking about some of the earlier versions being polystyrene coated, but what's the current sort of best conjugation technique? Is that still using the Sortes approach? Or?
Yeah, we're using probably not direct Sortes conjugation for our future project, but involving click chemistry, which is a bit cleaner because we can prepare both the particle and the antibody. We can prepare that separate and clean it up, and then just bring them together for the final conjugation step. But it will also involve Sortes for the antibody part. But the final assembly is likely click chemistry. And the coating is very important. The coating of the particles to avoid, a lot of people know, of course, that nanoparticles have the tendency to go into the liver and be quenched by the immune system. So to make them non-visible to the immune system is a big challenge for nanoparticle work.
Great. Well, thanks very much, Christoph. And it's probably a good point to note that the facility is not just available for researchers and discovery science work, but it's also available for industry partnership work as well. So we really encourage approaches from some of the biotech industries who are interested in adapting their current approaches to using these nanoparticle technologies with this new system. It's, I think, a really exciting new possibility for our commercial partnerships. Okay, so thanks, Christoph. We'll now move on to our third presenter in this symposium, Professor Vipul Bansal, who's director of the Sir Ian Potter Nanobiosensing Facility at RMIT University. He's the founding director of that facility, and his research encompasses sensors and diagnostics, biomedical imaging, cellular immunotherapies, microbial and wound management, and environmental remediation. He's the founder of NextGen Nanosensors, which is commercializing wearable UV sensor technologies to help protect from skin cancer. And his group is very active in the fabrication of biomedical imaging agents. So we really look forward to your presentation today. Thank you, Vipul.
Thank you, Gary, for that kind introduction. Let me start with congratulating Christoph first. Christoph, congratulations, that was a great news. And then moving on to my presentation, if we take the message from Patrick, I kind of gathered that there are two areas of scope in MPI. One is towards expanding the applications in contrast agent-based imaging. Another is development of new nanoparticle contrast agent for MPI. So we haven't done any work with MPI yet, but I'm going to share some of the work that we do with magnetic nanomaterials in terms of fabrication of magnetic nanomaterials. And also then I will share an example of a first in human study that we recently completed for tracking cellular immunotherapies in human using some of the dual MRI PET contrast agents. So our group is 50% focused on, half of the group is focused on material synthesis, and we have been doing this project for last 10 years. Precisely in 2013, the project started with an ARC linkage grant where we started making nanomaterials. So I will just show some of the capabilities and the requirement when it comes to biomedical contrast agent. So good quality iron oxide nanoparticles are typically synthesized in organic phase. As you see on the image on the left-hand side, here are some superparamagnetic iron oxide nanoparticles. They're synthesized in the hexane phase. It's quite challenging to transfer them into the aqueous phase. So what we have done now, you see the central panel. The one way of transfer to aqueous phase is potentially you can have a very small coating of silica around them. You don't want a very thick silica coating because that will affect the saturation magnetization or magnetism of the particles. So very thin coating of silica, we can control the silica coating. And also we have developed some patented methods by which we could transfer the particles into aqueous phase. As you see in the last panel here, particles were originally in the hexane phase. You could transfer it into the water phase, and you could reversibly transfer back to the hexane phase. So these were some of the capabilities in terms of aqueous dispersibility of the particles, and these capabilities are general capabilities that could be applied for many different type of particles and nanoparticles. The second thing that we try to control is the size and shape of the nanoparticles. I haven't shown the examples of size here, but you can see on the left-hand side image, the particles are quite monodispersed. They're probably around 10 to 50 nanometer here in size, but we can make them all the way to 2 nanometer to 500 nanometers, and while retaining their monodispersity. What we have been recently doing, and this is the data here on the 3 panels on the right-hand side. There's unpublished data, so I haven't provided much details, but we have been able to synthesize these 2-dimensional sheets of iron oxide. It's quite challenging to synthesize them because magnetic materials, they tend to form a spherical isotropic shape, but we can synthesize some of these materials in anisotropic shapes. As you can see, magnetite sheets here. You can also see, we can also control the composition. Here you have hematite cubes and some other magnetic materials like iron sulfide. Iron sulfide in itself is not that magnetic, but we could create some iron deficiencies into the system, and we can make this material magnetic. So this is also, you see some crumbled ball-like structures, crumbled structures in that rightmost panel, but they're very thin sheets of iron sulfide. The other trick that you can do to control the magnetic properties of these materials is to control the dopant species. So you can introduce a number of dopant species. On the left panel, you see an example of cobalt-doped magnetite sheets, the magnetite sheets that you saw in the previous slide. We have doped it with cobalt, in this case 2.5% cobalt. In EDX map, you see the cobalt signature and Fe signature coming from these sheets. On the right-hand side, you see, an example of a nickel-doped sheet in this case. So we have now actually 5 different dopant concentration of each of these transition metals. So 14 different transition metals and 5 concentration, almost 70 samples. We were going to run it last week through MPI scanner, but then we had lockdown in Melbourne. So we look forward to run some of these samples in MPI scanner and see what kind of performance these materials show. So now I will give an example of the work that we have been doing since 2013. This work was started through an ARC linkage with a local Melbourne-based company, Cell Therapies Pty Limited. And the target of that project was to develop some nanoparticle contrast agent that might be suitable for cell tracking application. So what is cell tracking or CAR T cell therapy? So we can isolate some T cells from the patient, for instance, and then you genetically modify the cells and you make them CAR T, which is chimeric antigen receptor T cells. Now, once you isolated these cells, you multiply them in the test tubes or petri dishes, And then basically you infuse these cells back into the patient after multiplying them, and you hope that these will target the cancer cells. So there are a couple of CAR T-cell immunotherapies already commercialized. One of them is Chimera, for instance. So once you put them into the body, now these are kind of genetically modified cells in a way. You also want to know where they are going. So cell tracking is quite important to see the efficiency of the cell, whether they're reaching to the targets or not. So they have commercial, there's a cell tracking capability has commercial as well as clinical value, because when pharma companies are investing in CAR T-cell therapies, they want to screen out at the early stage of the development, which therapy is less powerful and which is more. So what we did in this project now, the thing is, if you can somehow put some nanoparticles or bind some nanoparticles to these cells, and before you administer these cells into the patient, then there's a possibility to track them. But we are working with T cells, and biologists know that T cells are non-phagocytotic in nature, so it's not that straightforward to feed nanoparticles to these cells. But anyway, we were able to achieve it. So the nanoparticle that we designed for this project was a dual PET-MR contrast agent. So in the center, you see iron oxide nanoparticle SPON, around 12 nanometer in size, 12 plus minus 0.5 nanometer, quite a tight size range. And there's a very, very thin, around 0.5 nanometer thin silica shell around it. And the thing that we really did a kind of innovation in this project was, so generally when you bind a PET agent to a nanoparticle, it is bound through a ligand, a chelator. So some of the work that Peter Mac colleagues and clinicians were doing previously with PET contrast agent, one of the biggest challenges was the PET contrast agent used to leach out of the cells, and it used to affect the resolution of the imaging. So what we did in this case, we took copper-64, and then we made it into a nanoparticle form. So the radioactivity signatures that you're seeing around it is basically copper nanoparticles bound to the iron oxide nanoparticles very tightly, no covalent linkage, it's just directly embedded into the silica shell, but this copper-64 is in the nanoparticle form. So what's the advantages and disadvantages of going through this pathway of imaging? As Christoph already mentioned, means if you combine PET and MR, PET has higher sensitivity, MR offers higher resolution, so you can combine the best of both worlds. Also, the PET signal will fade away after some period of time because it has got limited half-life. Like in case of copper-64, it's 12.7 hours. So initial time points, you can potentially image through PET where cells are still dispersed. Sensitivity is more, but later on when the cells accumulate in a particular organ, then you could potentially use MR. So simple, scalable, and powerful cell tracking platform. This is readily taken up by cells with irreversible and strong binding, as I already said, because copper-64 is in nanoparticulate form, and we don't see any leaching. And we did studies where we looked at whether the radiation itself will have effect on the functionalities of these CAR T cells. So the low radiation dosage, which is 200 to 300 megabecs of PET radiation dose, doesn't affect the functionality of the cells. In terms of disadvantage, there's potentially additional cost in addition to the expensive cellular therapeutics. Additional consent process may be required for approvals, TG approvals, FDA approvals, and so on. And the thing is, because the half-life of copper-64 or radiopharmaceutical that you use is quite low, and because you have to make copper-64 in the nanoparticulate form, then you have to optimize the chemistry in such a way that it happens quite quick, because you need the radiologist at the hospital to manufacture these nanoparticles. So we kind of fabricated a kit where within half an hour, what they have to do is there's already a pre-existing kit supplied by us, and the radiochemist just add the copper-64, which comes from Austin, and within half an hour, you have the PET-MR contrast agent ready without requiring any centrifugation and purification, which is not available in hospitals generally. So all of these things over the last 10 years, we have filed 15 patents, and there's a lot of IP in different aspects of this project. So this is one of the data showing in animals, for instance, co-registration of PET and MR signal. In this case, we are looking at intravenously injected HER2 positive CAR T cells. That was the initial work done by a PhD student, and we can see the CAR T cells have localized, in this case, in the tumor, which is evident from both PET and MR signature. One of the positives from this aspect was we didn't see any signal coming from brain and heart, which gave us confidence that these particles do not dislodge away from the CAR T cells once they are in vivo system. And this image is on a 3 Tesla MRI machine, so this is on a clinical scanner. So if we move on to the human trial, there were some interesting observations. So what you have here, let's start with the text on the right hand side. So first thing is copper-64 labeling efficiency. So I already mentioned about it. So once you receive the copper-64 in the form of copper chloride salt in HCl, you need to be able to radio label your iron oxide particles quite quickly. So we were able to achieve more than 99% radio labeling efficiency, which means you have very small amount of free copper-64 there. So you can directly inject that dose into the CAR T cells. The second thing is, once you have this PET MR contrast agent ready, then it needs to be able to label CAR T cells, because you don't want a lot of free contrast agent in your soup or the cells that you're going to inject into the patient, because clinical protocols would not allow you to remove the free particles from the cells, for instance. That complicates the process. So what you see in the table now are 3 different patient runs, where we took the fresh cells from the patients versus some of the cryopreserved cells which were donated previously by the patients. So what is interesting is the labeling of the nanoparticle labeling efficiency in the cryopreserved cells is almost twice as in the fresh cells. We still do not fully understand this reason, but one of the potential reasons may be because there was DMSO in the cryopreserved cells, if that is somehow increasing the efficiency of the loading of nanoparticles on the CAR T cells. We're still trying to figure that out, but we are not sure yet. The next thing was CAR T cell viability. So what we looked at is whether these cells were viable after having these copper-64 and MR contrast agent. So post-labeling viability immediately after the labeling was more than 90%. And we also looked at what happens after the 4 hours, because once you administer these cells into the body, you need to look at what's potentially the long-term viability. So within 4 hours, they should start biodistribute. So we looked at 4 hours viability. There's not much influence on the viability. And the number of replicates here are not too large, because we should appreciate that each of these experiments costs $100,000, because the cells gets collected through apheresis process, and also so it's quite expensive to run these experiments. So whatever the clinical requirement was, those many experiments were run to validate the experiments, the results. Now, once you have loaded your particles of contrast agent into the cells, next thing you have to look at whether these CAR T cells retain their functionality. So what we're looking at here is the cytotoxicity assay, which means the ability of these cells to kill other cells. That's what you want, kill tumor cell. So basically in all these graphs here, the red ones are the cells, Lewis-Y CAR T cells with nanoparticles. The black ones are without nanoparticles. What you are seeing in all these figures, the black and the pink are overlapping, which means that nanoparticles are not affecting cytotoxicity as in the first panel, and also the production of cytokines such as IFN gamma and TNF alpha, which means these T cells, they retain their functionality post treatment with the nanoparticles. So now we will see some of the human data. I've put the data for 2 patients here. So this is the data after 4 hours of infusion of these labeled cells into the patient. Because we're looking at 4 hours, we are really not going to get the MR signal. And these patients were administered at Peter Mac, and then because Peter Mac has only PET and MR machines are separate, but at Royal Children, we have a dual PET MR machine. So there were logistic issues to move every time patients from Peter Mac to Royal Children, for example. So at 4 hours, we only looked at the PET data. So what's interesting, and here we are looking at Lewis-Y CAR T cells model, and these are advanced stages of tumor in these patients with acute myeloid leukemia, but without any extramedullary disease. So the tumor was metastasized everywhere. So what we see, there is a moderate uptake of these labeled CAR T cells in lungs, but this activity doesn't increase, as we saw from the other images, over the next few days, suggesting that it was a transient process. And also we saw some activity in the liver and spleen, suggesting that the margination of the cells to the RES, reticuloendothelial system. Now, if you look at a bit of long-term data, say 22 hours and 46 hours, so these are dual PET-MR scan on one of the patients. So as you can see on the left-hand panel, which is the baseline, where I highlighted this tumor, there's not really an increase in activity of the PET agent over a period of time at day 1 or day 2 in the tumor. So what does it suggest? But at the same time, also, there is no activity in the brain and the heart as well. So having no activity in brain and heart is good, which means that the clinical trial agent the contrast agent is safe, but also which means that this potentially means that these cells are not reaching to the target. But we will see more about that later on. So this is an unproven therapy. This was a therapy, this why therapy under development. So it's not a commercial therapy. So we're looking at the capability of this therapy for treat tumors. So our data shows that the therapy is not reaching to the tumor. Now, we have some long-term data. As I said, means the advantage of having PET-MRI dual contrast agent is you could have a long-term data on the MR signal. So in terms of MRI signature, after 1 day, we see a 40% reduction in contrast, which more or less remains same over a long period of time in the organs where we saw the MR and the PET co-registering. So as a summary, this first-in-human study provides insights and valuable visual data into individual response to adoptive cell therapy, such as CAR T cell therapy in this case. This PET-MR cell tracking system allows us to determine how many labeled CAR T cells distribute to solid tumors and various other body organs using high-fidelity imaging. So we've done some calculations where we can now calculate how many CAR T cells have reached to which organ as a function of time. This powerful technology may offer insights into cell therapy target validation, as I mentioned, because for pharmaceutical companies, it's quite important to get this information early on. Target validation, biodistribution, product dosage, and optimum route of delivery, with view to improving the safety, potency, and tolerability of the cell therapies. So no cell tracking to tumor sites in humans has been identified to date. It's possibly either due to the CAR T construct itself or the tumor type that we evaluated in this case, or potentially because of suboptimal CAR T cell migration to tumor site. So these therapies, if we had the clinical data of the recovery of the patient, these therapies did not work into the patient. So basically our study validated that potentially these CAR T cells are not reaching to the target. So next step is to incorporate this technology into a clinically proven CAR T cell therapy. Because of the confidential nature of this, non-confidential nature of this forum, I can't tell much what we are doing, but we are going to try it in some of the proven commercially available technologies. Now that's what we're working on. Of course, a large number of people were involved in this work. Cell Therapies, PT Limited, they are our industrial partner on this work, and the principal clinical investors are Simon Harrison and Bansal at Peter Mac. Cyclotech got involved in the ARC linkage originally, Cell Therapies and Peter Mac were involved. Then what happened then, we got subsequently a Global Innovation Linkages grant to do the first clinical trial, where Cyclotech got involved. They had a radiopharmaceutical manufacturer in Australia. So they helped in translation of the nanoparticle synthesis in a clinical environment. And then a lot of other people involved at RMIT University, a number of students who are now postdocs, and some of the other colleagues have also contributed to this work over a period of time. And last but not the least, I would like to thank the patients and the families who actually participated and made this work possible. And thank you all for your attention.
Excellent. Thank you very much, Vipul. That was fascinating. Terrific work. Really impressive.
Thanks.
Can I ask you about the early work you showed looking at some of the different, if you like, geometries of nanoparticles? We're talking about controlling the size and shape of nanoparticles. And then you talked about some of the sheets and also about the doping of nanoparticles and your interest in, testing I guess the sensitivity to those doped nanoparticles. Can you give us an insight into what your thinking is behind those strategies?
So in terms of dopant species, there have been publications in the literature where they've shown, for example, iron oxide has around 60 to 65 EM per gram saturation magnetization value. If you dope cobalt and manganese into it, you can bring it to 250, 4 times higher saturation magnetization value. So that's the strategy around it. In terms of morphology, we are seeing some very weird results, if I may say. So in case of those iron oxide sheets, for instance, which is again iron oxide, and theoretical values of iron oxide is around 90 mg per gram. Now we are kind of approaching that theoretical limit in terms of those iron oxide sheets. And the reason may be because now these are few nanometer, 1 or 2 nanometer thin sheets and quite long, up to micron, if you may like. Now magnetic domains may be restricted. I don't know, I'm not a magnetism person really, but we're seeing some very interesting results. It might be much easier for the magnetic domains to be oriented in those kind of structures than in spherical particles where they might be just randomly oriented. So it would be interesting to see how they behave in an MPI scanner.
Yeah, fascinating. I have another question around your use of copper-64 and the fact that it's a 12-hour half-life. There are other isotopes such as zirconium-89 which could take you out a lot further and track that. I just wondered if that's something that you're pursuing as well, or do you think that you can get enough MR contrast with the SPION out at 3 and 5 days?
So that's an interesting one. So the original ARC linkage had copper-64 and ZR-89 both. ARC gave us half of the money, so we did copper-64. And we are now putting in application to do ZR-89. That we would really like to go to ZR-89. And what we learned from our clinical trial results that we have to increase our iron oxide dose by 4. So we use 6 milligram iron oxide per patient. That's how much was administered overall in a patient. To get a proper contrast, we need to inject 24 milligram equivalent. So we need to change our SPION contrast agent, make it more magnetic, or increase the dose. And then also we have to change copper-64 to ZR-89 to make it a better contrast agent.
Yeah, fascinating. Really interesting to watch this work come through. Vipul, thank you very much.
Thank you, Gary.
Impressive. So now we'll move on to our fourth speaker in the symposium. It's a pleasure to introduce Dr. Karen Alt, who's an NHMRC MRFF Career Development Fellow, head of the Nanotheranostics Laboratory at the Australian Centre for Blood Diseases at Monash University. Karen's research focuses on the development and clinical translation of diagnostic imaging and therapeutic agents for early diagnosis, therapy and treatment monitoring in cancer and cardiovascular diseases. She's developing novel theranostic approaches, and I know very interested in the hyper capability. And we look forward, Karen, to hearing your presentation.
Hi, everyone. Thanks for having me. So nanotechnology is defined as the intentional design, characteristics, and production, application of material production, devices, systems, control sizes, shape, and the nanoscale ranges. The combination of therapy and diagnosis called Theranostics, and it has been seen quite often so far with a small peptide against prostate cancer, which the PSMA was labeled to gallium-68 for diagnostic and or to lutetium-177 for therapy. This is wide in clinic, even here at the McCann Cancer Centers. Okay, so as I said, so far we have seen Theranostic as combined radio tracers, one for therapy and one for diagnostic. which is done here at the Peter MacCallum, for example, for prostate cancer, and of course worldwide. However, a new concept of Theranostic I'd like to present today, a Theranostic platform, which is non-invasive, safe, provide full real-time feedback with therapeutic function. You can see here 2 pictures of our Momentum installed downstairs. So it's our first Theranostic synergetic modality, MPI and Hyper. So the MPI as a diagnostic tool paired with the Hyper, which is a localized therapy guided by the MPI image. And the MPI could be employed to monitor therapeutic responses and enable personalized medicine. Of course, we have various applications for this system, for example, in vivo imaging, vascular imaging, perfusion imaging, multicolor imaging with different sizes, shapes, and of course, temperature guided therapy, as I mentioned already, like hyperthermia therapy, compared maybe even with chemotherapy. We have multimodal imaging like MRI with MPI, MRI with PET, MPI with PET, even cell tracking like CAM cells or TCAR cells. However, even this application seems to be endless. My presentation time is restricted, so I will focus only on cancer applications in the current presentation. And successful application of MPI for imaging of cancer depends on the nanoparticle accumulation at the tumor site at sufficient level relative to other sites, for example, liver. We have seen already a couple of successful examples. Here, one you et al. Synthesized the 1 octotant packet-coded nanoparticle in MPI tracer to achieve optimal resolution and blood circulation half-life. Due to the quantitative nature of MPI with zero depth attenuation and non-invasive nanoparticle biodistribution through time is possible. Du and team tested the EPI effect of their tracers on MPA performance with regards to half-life and tumor being rats. The blood circulation half-life was calculated by roughly 4.5 hours, and a tracer accumulation showed in the tumor being rats via passive targeted tumors with a high signal-to-noise ratio. So the dynamic of the MPI showed a maximum reach in the tumor at 6 hours with a full clearance after 48 hours. However, targeted ligands for MPI could further enhance tumor contrast and allow intensive differentiation form to normal tissue. So Ami and team conjugated lactoferrin, a brain cancer targeting peptide, to nanoparticles and used with and without an external magnet and with and without targeted motif to evaluate the EPR-mediated accumulation to improve the localization of the nanoparticles to the tumor xenograft model. Using the lactoferrin showed an accumulation in the xenograft animal model as indicated by the MPI signal, and by placing a magnet close to the surface to the tumor, a superior accumulation was observed, which offers an alternative strategy. So here, let me please to introduce first the team who created this results. That is Jury, my talented PhD students, and Robert, our RMIT facility manager. Both of them created these results done in our facility downstairs. We tested new nanoparticles from the company Egan, and get kind of used to the software and used to the system itself. We can show that our nanoparticles have a linear relationship of the ion to the MPI intensity as described in the literature. So any quantification analysis of ion at region of interest is possible. And I loved actually Patrick's saying that the particle giving a bow tie, because that's a perfect description, was our particles as well. So here, this particle was injected into an animal, healthy animal, and circulated for 30 minutes. And we took different time points in the MPI image. And as mentioned previously, due to the quantitative nature of the MPI with zero depth attenuation and non-invasive nanoparticle biodistribution through the time is possible, and a direct correlation between ion accumulation and MPI can be done, as here shown in the liver. So what is next for us? Next for us is hot out of the fax machine. So we were able to target our nanoparticles to an antibody against receptor 2. And we could show that the targeted particles have a binding as strong as a non-targeted antibody to the target, which is absolutely amazing. And our xenograft animal model is sitting downstairs and just needs a bit to grow before we can scan in xenograft. So, as I mentioned previously as well, we have a synergetic modality. So based on the MPI image, we can do image-guided localization therapy with the Hyper system, which is one of the advantages of our setup. Here is a cool picture that is a pre-setting, and unfortunately, it will be covered at some point, but I really like the outfit right now. So what makes hyperthermia so attractive as treatment option? It is a typical goal to heat a target tissue region to roughly 42 degrees to achieve the desired effect, which is heat-induced cell death. An ideal theranostic workflow in vivo has been demonstrated by Tai and team. MPI-based images guided therapy. The MPI image was shown both tumors implanted in a xenograft animal model, one on the top and one on the bottom. Based on the MPI image, highly localized heat dose only to the bottom tumor was applied while sparing nearly healthy tissue that might have non-specific accumulation of nanoparticles or body natural clearance organs of the nanoparticles such as liver or spleen. These clearance organs are sensitive to heat damage, which has been shown previously. So localized heat was monitored by 3 temperatures probes, which were placed in the tumors and in the rectum, shown in the middle image. And on figure 3, you can clearly see a reduction after 3 days on the bottom tumor where the heated was applied to. Another example is Du and team. They improved the delivery uniformity by targeting a tumor with an 18 nanometer iron oxide particle targeted to a peptide cracker, which improves the delivery of the nanoparticle to the tumor. In vivo anti-tumor magnetic hyperthermia therapy effect was evaluated. Data showed that the improved targeting and delivery uniformity enables more effective magnetic hyperthermia cancer ablation than other identical non-targeted nanoparticle and all the other controls. Shown here once as a result of the fluorescence and once as a tumor size. So far, we tested our particles to their heating pattern in different volumes and concentrations, and always in mind, we want to have roughly a temperature of between 42 and 43 degrees. So with our particles, we would need 25 microliter of 0.25 milligram to reach a temperature of 46 degrees, or 50 microliter of 0.5 milligram iron oxide solution, which comes roughly to 40.6 degrees. But this can always change in vivo, and we have to see how these particles actually we have in vivo. In addition, the therapeutic technique requires increased temperature by only a few degrees, for example, by limited or mild hypothermia in conjunction with chemotherapy or the radiation therapy, which has been explored as a way to enhance therapeutic potential. And yeah, I'm really excited to go down this pathway and yeah, do more studies. So the key applications for localized hypothermia are direct tissue ablation, which I gave you examples for, either with targeted or non-targeted motif. Heat-sensitive gene activation sounds like a very interesting field, as well as heat-sensitive drug delivery. As a very hot topic at the moment is the immunogenic tumor stimulation from cold to hot tumors, which will improve the infiltration of tumor, which will increase the inflammation of the tumor with T cells to fight the tumor, or even the combination of 2 or 3 of the application will be the recipe to fight cancer. So overall, I hope in summary I could convince you and give you a quick overview how great the system is and how well they can work together. No doubt it will play an important role in personalized medicine in the future. However, time for nanoparticle development with regards to non-targeted versus targeted, without a drug, size, shape, charge, just to mention a couple of alternatives and changeable parameters is needed. And yeah, I think we are really proud that we are the first in the world of the combined systems. And I would like to take the minute to say thanks to the team, especially to Yuri and Robert. Without the preliminary data would be not possible. Of course, Magnetic Insight and Plant Bio, which have been amazing so far, Gary and his team, and of course, our funding partners. Thank you.
Fantastic. Thank you so much, Karen. That's really exciting to see that first data coming out. And I had a question about your hyper study, or at least perhaps the published work you showed. Was that in an animal model? Were they able to achieve that many degrees localized temperature increase in like up to 5 minutes hyperthermia?
So we were able to, depending on iron oxide and the volume, to get to up to 56 degrees within 5 minutes, yes.
Wow.
But that's all in vitro, not in vivo.
What's the temperature increase, do you think, in a typical, say, a mouse model study?
Usually you would time it to 42 degrees.
42, yeah, within 5 minutes typically.
And that depends, again, if you have a chemotherapy attached or radiotherapy attached or just heat therapy.
Yep, that's terrific. I noticed your bow tie pattern in your image, but I noticed a different orientation. It made me wonder if there's any sort of down under effect on that.
Oh, you can turn it around. It's how you want to have it.
Okay, great, thank you. Well, if you want to stop sharing your screen, we do have we do have some questions. And firstly, I think we've got one question here for the panel. What I'll do is invite our symposium speakers to turn their videos and audios back on, and then we can address a few of the questions that have come in. First question that's come in, open to any of our presenters, is could we say that MPI could replace other imaging technologies such as PET-CT in the future? Is that reasonable? What are your thoughts on that, Patrick?
I don't think it would replace PET-CT. I like to use the example of MRI and CT. We use MRI to image soft tissue. We use CT to image bones. MPI, the applications that we're going to be using it on, I think a lot of them will be adoptive cell therapies and things where we're looking at blood perfusion, the stuff that Christoph is working on, labeling like thrombus. And so it's just going to be a very different application than what we use PET-CT for.
Any thoughts from other panel members? Vipul, do you have a view on that? Do you think that MPI is going to supplant PET, or it's just really in combination?
I agree with Patrick. Actually, it's too early at this stage to say, because the way instrument is designed, there are bulky magnets there. So the first step would be to try to image a whole body, whole patient. And once that is achieved, then we can think about having it replaced with some other technologies which are already there in the hospitals.
Okay. There's another question that's come in from Lars Esser. He's got a question about the hyperthermia application. Karen, if you know what is the localized heating, i.e. within the 10 to 20 nanometers of an iron oxide nanoparticle surface, what in that sort of very direct localization of the particle, is the heating at the temperatures you're quoting, or is it much higher localized? Do you know? Is there a heating profile, for example, as you come away from the nanoparticles?
So far, we have seen, or literature says, everything between 18 and 25 nanoparticles we have seen with completely different heating patterns. Even if the particles are PEC, for example, PEC coded, they have a different heating pattern. So I assume if you have a particle, ship it over and we're testing it.
Mm-hmm. Do you know, Patrick, anything about the profile of the heating increase around the localization of the nanoparticles?
So I actually asked this one of Carlos Rinaldi a number of years ago, and this is a little bit hearsay, but from what I understand is that the way the thermodynamics works is that there's not a really high temperature right next to the particle. It really gets spread out from the way the heat diffuses. That the temperature right next to the particle is pretty much the same temperature, say, 10 microns away or something.
Mm. Okay, so it's really at body temperature. This is thermally distributing quite rapidly. At least
yes. So essentially, there's no nanoscale heating profile. It's really the micro scale.
Mm. Yeah. Okay, that's good. Now, happy to invite our panel members to further add to any of their comments, or perhaps ask other presenters any questions if they have them. Be interested to generate a little bit of discussion around some of these presentations.
I have a question for Patrick in regards to hyperthermia. So the way MPI hypersystem work, is it similar to the traditional hyperthermia? It's just that MPI is able to focus the fields in the zero field region, or whatever you may like to call it. So it's just localized hyperthermia, but otherwise the concept is same, or is there any difference?
That's correct. It's the same, except for the addition of the selection field. Now, something interesting about the selection field is that it can change in strength and in location. And so the system that you guys have, so you have the animal, the physical location of the field-free region, you can change the size of it by moving these two magnets, and the magnets will move up and down and left to right. And then the animal can move forward so that you get 3 axes of movement. And then it can change the strength so that it fits almost the whole animal for the heating, all the way down to, I think about 4 millimeters or so for the sharpness point.
All right, thank you.
Yeah, I think there's going to be a lot of interest in trying out the hyperthermia capability. And as Karen mentioned, what we saw there was, I think, an early version of the instrument, Patrick, and there's a more compact version en route. Is that correct?
So it's still in development. So it might be a bit of time before that one's ready, but it's still in development.
Right.
Patrick, I had another question for you around the opportunity for looking with what's called color MPI. I don't think that's been mentioned so far today. My understanding is that's going to enable us to look at different nanoparticles, different sized nanoparticles in vivo simultaneously. Be interested if you could, speak briefly about the potential of color MPI.
Sure. So color MPI We use it as a way of describing really where we're using the relaxation properties of a nanoparticle to add an extra layer of information to the image. And so a couple examples are having two different types of nanoparticles, each with different relaxation properties, and differentiating between them. So that's one way of looking at color. Another way of looking at color is a single nanoparticle, and then it has some interaction with its microenvironment. And you're able to see, you know, say if it's binding to something or if it's bound in a nano construct, and you're being able to differentiate between bound and unbound or other, you know, viscosity or other things like that. So that's the way that we use the phrase color MPI. And I would say it's very much under development. What we have is a software development kit that you guys will soon get, where you're able to really explore that. So we give example code that does differentiation between two different colors, but of two different types of nanoparticles to get two different colors. The differentiation is, there's some false positives and false negatives and stuff like that. And so we give you the base code of it, so that if you guys are developing, you know, new types of nanoparticles that can improve the differentiation, or mathematical approaches of improving the differentiation, that you guys have all of the you know, framework to be able to build that with. So we're quite proud of the SDK in its ability to let people really look at the engineering behind how the technology works and be able to actually make fundamental contributions to the technique. I think it's going to be a lot of very great papers coming out of something like this.
Excellent. Yeah, that's really exciting. Okay, do we have any other questions from our audience or any other comments from our panel presenters? Okay, I think we've probably then come to a close in our launch event today. So it's been really great. I've just enjoyed listening to this science, and particularly seeing those first results, Karen. I know that's been some pretty late work over the last week or two, so terrific job. Thanks very much, Vipul, for your presentation and for RMIT's support for our facility and collaboration. Thanks to Trent and to Trend Bio for your great support of the installation, and I look forward to working with yourself and Magnetic Insights in our MPI Centre of Excellence. That's going to be terrific. Thank you very much to Christoph and also to Robert and Kylie, and particularly thank you to Merrin Morrison, who's behind the scenes here, has made our event run very smoothly today and really ensured that this went off without any hitches. So thanks to all our speakers. Thank you for attending our event today, and please make contact with Rob around having a look at visiting the Arambi site, having a look at the facility. You can find information about accessing the MPI facility on the MBI website. It is open to researchers, and in partnership with companies, we can also provide access to the facility. So we really encourage and reach out to all interested users and collaborators on use of the facility. So I think that with that, I'll bring it to a close. Thanks for your attendance, and we look forward to this hopefully being the start of some periodic sessions and symposia on MPI. Thanks, everyone.